Hello guys, welcome back to my channel. So today we will continue with our next transmembrane binding receptor, that is Jak-Stat binding receptor, and next we have the nuclear receptors. So let's start with the transmembrane Jak-Stat binding receptors. Let us assume that this is our cell, and on the surface of this cell or in our cell membrane here we have a receptor. This receptor is tyrosine kinase associated receptor. So whenever a ligand binds to tyrosine kinase associated receptor, this receptor will get activated and it will achieve intrinsic tyrosine kinase activity. Previously, this receptor did not have any intrinsic tyrosine kinase activity, but just after interaction with the ligand, it gets activated and it gets some intrinsic tyrosine kinase activity. So after they have been activated, it will undergo dimerization. So while it has got dimerized, there is a special kind of enzyme that is present known as genus kinase. This genus kinase, also known as shortly JAK, will go and bind to the receptor in a specific place and they will cross phosphorylate the tyrosine residue that is present in the receptor. Cross phosphorylation means this JAK molecule will phosphorylate this tyrosine residue and this molecule of JAK will phosphorylate this tyrosine residue. So after they have done the cross phosphorylation, they will interact with a protein that is STAT. STAT means signal transducer activator of transcription. So what STAT will do is they will transduce a signal which will lead to activation of the transcription process. So STAT, it will go and interact with JAK. After they have interacted with JAK, they will show some action. They will carry some signal to the nucleus. For that, they have to detach from the JAK protein and they will go to the nucleus and they will interact with the nucleus, and there they will start the process of transcription and then followed by translation. So the substances that follow these pathway is prolactin, interleukin 2 and some other substances which follow this Jak-Stat binding receptor pathway. Since Jak and Stat they interact with each other and carries this pathway, therefore this receptor pathway is known as Jak-Stat binding receptor, and this is also one of the one pass receptor that is present on the membrane of the cell. Now, one of the point that we need to know is the difference between the tyrosine kinase associated receptor and the tyrosine kinase receptor that we have seen in our previous video is that tyrosine kinase associated receptor do not have any intrinsic tyrosine kinase activity initially. Like in normal state, when there is no ligand attached to it, it do not have any intrinsic tyrosine kinase activity. Whereas in previous video we have seen that in case of tyrosine kinase receptor, they had tyrosine kinase activity from the very beginning. Also, in case of tyrosine kinase associated receptor, they do not have any link to JAK unless and until the ligand is bound to this particular region. Whenever this ligand is activating the receptor, JAK comes and binds to this receptor and JAK gets activated, and then again in turn STAT interacts with JAK and all the following processes occur. But JAK also being a tyrosine kinase protein, it do not have any relation with receptor initially. It only gets activated or attached to this receptor because of the ligand that is binding. So this is a difference between the tyrosine kinase associated receptor, where it is just associated, whereas in case of tyrosine kinase receptor, there was tyrosine kinase present in the internal domain of the receptor. It was a part of that receptor. So this is a basic difference between these two receptors. Next, we have type of nuclear receptors. So for nuclear receptors, we have seen in two cases where they are transmembrane receptor, meaning that they are present in the cell surface or the cell membrane. But nuclear receptors, they are present inside the nucleus. So we know there are substances that are lipophilic in nature and some are hydrophilic in nature. So for the substances that are lipophilic in nature, they can easily penetrate the cell membrane and they can go inside the cell. So for those substances, receptors are present either in the cytoplasm or in the nucleoplasm. So the ligands like steroid derivative, estrogen, progesterone, glucocorticoids or vitamin D, retinoids, they are the ligands that penetrates the cell membrane and go and interact with this nuclear receptors. So this is our nuclear receptor that is present in the cytoplasm. Since a receptor is a peptide, so it will have an amino group and a carboxyl terminal. In this case of receptor, there is a domain where the ligand will interact and there is a domain where DNA will interact. But when this receptor are not in their active form, there is a protein that is binding or protecting this DNA interacting domain. So this protein is known as heat shock protein. This heat shock protein protects the DNA binding site. Normally, without the ligand, this heat shock protein is protecting this DNA binding site. So whenever a ligand come and bind to this receptor, what will happen is the heat shock protein will move away or it will detach from the receptor and this DNA binding site will be exposed. This exposed site, the receptor along with the ligand will go to the DNA and it will start interacting with the DNA. Now, the site of the DNA where this DNA binding site is interacting is known as the enhancer region. This region is present in the DNA. And this domain will interact with this enhancer region, and on enhancer region, it will stimulate the formation of RNA from DNA. So this process is known as transcription. So when mRNA is formed through the process of transcription, next will be formation of new protein, that is translation process will be carried out. So whenever new proteins will be formed, the response will be seen. So this was about the nuclear receptor and how this receptor is working. So let's summarize again that for the lipophilic substances, they can easily go into the cell and they will act as ligand and they will interact with this particular receptor. So this receptor, when the ligand is not present, they have the heat shock protein covering the DNA binding site. And whenever ligand come and attaches with the receptor, the heat shock protein will detach from the receptor and this DNA binding site will be exposed. Now this receptor along with the ligand will go translocate themselves and they will interact with the DNA. So this place where they are interacting with DNA is known as the enhancer region. So this DNA binding region or domain, it will interact with the enhancer region and they will start the process of transcription. After they have started transcription, then they will the process of translocation and ultimately they will show their responses. So this is about the nuclear receptor. So that's it for today. I feel that this video was helpful for you. If it was, then please don't forget to subscribe to my channel and let's meet in the next video. Thank you.